The first monomer that we're going to look at are nucleotides. Okay, so these make up both DNA and RNA, and they have similar but slightly different structures, which allows us to distinguish, for instance, a DNA nucleic acid from an RNA nucleic acid. And so that's all in the monomer. And so the base of a nucleotide, so that is the monomer of these nucleic acids, is a ribose sugar. Okay, so, and more specifically, this is a ribofuranose. Right? So remember, furanose means that this is going to be a 5-membered ring. And it takes this structure where CH2, and then we have a phosphate group. Okay, and note that the phosphate will be charged, okay? It can carry a -2 charge on the phosphate. Okay, so we have what's called a nitrogenous base that's attached to the ribose. And if we look at the numbering system, the numbering for this is where our anomeric carbon would be. So this is carbon 1, here's carbon 2, here's carbon 3, here's carbon 4, here's carbon 5, okay? So we have our anomeric carbon here. But instead of an OH group, we have a nitrogenous base, which we'll get to in a second, about what the structure of that is. So we have a nitrogenous base here, and so what we also have is a glycosidic bond between the ribose sugar and the base, okay? So back up a second. So again, this is ribofuranose. Okay. So the features we need to look at are, one, we have the sugar. So the ribofuranose. 2, we have the base here. And then 3, we have the phosphate. Okay. And so the phosphate, the base is attached to the 1 carbon, and the phosphate is attached to the 5 carbon. All right. So now this, again, is what you would find. This would be the monomer for RNA. All right. Now, for DNA, the nucleotide is very similar, but just slightly different. So first, DNA, right, stands for deoxyribonucleic acid. So this will be made of deoxyribonucleotides. All right, and so the structure is essentially the same. At the base, at the anomeric carbon, but at the 2 carbon, there's no OH group. And so that's why this is known as a deoxynucleotide. Okay. Otherwise, everything's the same. The general structure is the same. Okay? And so these are deoxyribonucleotides. Again, the important distinction is that at the 2 carbon, you can also think of it as the same as a 2-deoxy ribonucleotides because the OH group at the 2 carbon is missing compared to the ribonucleotide. Okay? So this is the base structure for these. And then what changes then when you're looking at the genetic code is the structure of the base.